Good day, and thank you for standing by. Welcome to the Celcuity Phase 3 VIKTORIA-1 topline results investor call. At this time, all participants are in a listen-only mode. After the speaker's presentation, there will be a question-and-answer session. To ask a question during the session, you will need to press star 1 and 1 on your telephone. You will then hear an automated message advising your hand is raised. To withdraw your question, please press star 1 and 1 again. Please be advised today's conference is being recorded. I would now like to hand the conference over to your first speaker today, Brian Sullivan, CEO and co-founder of Celcuity. Please go ahead.
Good morning. Thank you for joining us today. Let's now turn to slide 2. Before we begin, I must point out that some of our comments today contain forward-looking statements that are subject to risks, uncertainties, and assumptions. In particular, our expectations around gedatolisib are uncertain and subject to change. Should our expectations fail to materialize, or should our assumptions prove to be incorrect, actual company results could differ materially from these forward-looking statements. The description of these risks, uncertainties, and assumptions is included in our SEC filings. Let's now turn to slide 3. Today is an important day for patients who have HR-positive, HER2-negative advanced breast cancer. The clinically meaningful improvement in both progression-free survival primary endpoints from the PIK3CA wild-type cohort of Phase 3 VIKTORIA-1 trial was unprecedented in this disease setting. Dr. Igor Gorbaczewski, our Chief Medical Officer, will be joining us today to present these exciting results. We're also pleased to have Dr. Rachel Lehman, Professor of Medicine at the MD Anderson Cancer Center, join us to provide an overview of the treatment landscape for HR-positive, HER2-negative advanced breast cancer. And Eldon Mayer, our Chief Commercial Officer, will provide a brief update on our preparations for a potential launch of gedatolisib should we get FDA approval. Let's turn now to slide 4. We founded Celcuity because we believed that a functional analysis of the signaling pathways in tumor cells could yield important insights that weren't available from genetic analysis alone. When we turned our attention to the PI3K-AKT-mTOR, or PAM, pathway about 5 years ago, I couldn't reconcile two facts. First, the PAM pathway has long been recognized as one of the most important cancer drivers. And second, at the same time, very few patients were benefiting from drugs that targeted this pathway. It made no sense to me that there weren't any blockbuster PAM inhibitors. And we learned that the primary reason why PAM inhibitors have had limited impact, despite the importance of the PAM pathway, is because it is very difficult to safely and efficaciously inhibit this pathway. And there's multiple components, each of which need to be inhibited to comprehensively blockade the PAM pathway's activity. Otherwise, if only a single component is targeted, adaptive resistance arises and pathway shutdown is limited. Further complicating the challenge of drugging this pathway is the narrow therapeutic window that exists. There is a graveyard of drugs attempting to inhibit all these components that were not efficacious, too toxic, or both. The failures of these early-generation PAM inhibitors led drug developers to target single components, such as PI3K-alpha, AKT, or mTORC1, as a strategy to avoid toxicity. However, by compromising on the biological imperative that requires comprehensive blockade of this pathway, the resulting drugs have typically only demonstrated efficacy in patient populations that have pathway mutations. Eventually, we developed two hypotheses that led us to gedatolisib. First, we didn't think the pathway's relevance as a cancer driver was a function of the presence or absence of a mutation. It became clear, at least to us, that PAM pathway's role as a cancer driver and its overall importance was independent of any specific pathway variants or mutations. And this meant that an inhibitor that could comprehensively shut down this pathway could potentially be efficacious in patients lacking common mutations, such as PIK3CA. Second hypothesis was that a highly potent drug that avoided overexposure in key organs, such as the liver and GI tract, could potentially limit the incidence of the side effects associated with this pathway, such as hypoglycemia or diarrhea. Let's now turn to slide 5. And so today, we're very thankful that the data from the PIK3CA wild-type cohort of our Phase 3 VIKTORIA-1 clinical trial has validated our hypotheses. In patients with HR-positive, HER2-negative, PIK3CA wild-type advanced breast cancer, gedatolisib plus fulvestrant and palbociclib, and gedatolisib plus fulvestrant met the study's two primary endpoints by demonstrating statistically significant and clinically meaningful improvement in progression-free survival, or PFS, versus fulvestrant. Median PFS for the gedatolisib triplet was 9.3 months, compared to only 2 months for fulvestrant, a 7.3-month incremental improvement in median PFS. The hazard ratio was 0.24, which translates to 4.2 times higher likelihood of survival without disease progression than fulvestrant. Median PFS for the gedatolisib doublet was 7.4 months, again compared to only 2 months for fulvestrant, a 5.4-month incremental improvement in median PFS. The hazard ratio was 0.33, which translates to 3 times higher likelihood of survival without disease progression than fulvestrant. Let's now turn to slide 6. I have to say, we were genuinely amazed at these results. They're unprecedented and establish several new milestones in the history of drug development for HR-positive, HER2-negative advanced breast cancer. First, the hazard ratios reported for both the gedatolisib triplet and doublet were the most favorable ever reported by any phase 3 trial, first line, second line, or third line in this population. Second, the incremental improvements in median PFS for the triplet and doublet were the highest ever reported by any phase 3 trial for this patient population, receiving at least their second line of therapy for advanced disease. And third, gedatolisib is the first PAM pathway inhibitor to achieve positive phase 3 data in patients who have PIK3CA wild-type tumors and whose disease progressed on or after treatment with a CDK4/6 inhibitor. In light of this data, we believe the gedatolisib triplet and doublet each have the potential to establish a new standard of care for these patients. Let's turn now to slide 7. We're obviously very encouraged by these results and the implications for our now enrolling phase 3 first-line trial and our other potential future clinical development plans. Since gedatolisib has the potential to address a number of additional unmet needs in a variety of additional settings, we're thankful that our intellectual property position is very strong. With a recently issued patent, we expect to have patent exclusivity with gedatolisib until 2042. Let's turn now to slide 8. It's my pleasure to introduce Dr. Igor Gorbachevsky, our Chief Medical Officer. Igor joined us at the outset of our clinical development program for gedatolisib. He's done a fantastic job of building and leading the clinical development team that has delivered these trial results. Igor, you're up.
Thank you, Brian. Let's turn to slide 9. We all know that most common type of advanced breast cancer is hormone-positive, HER2-negative disease, and approximately 60% of patients will have PIK3CA wild-type breast cancer. Unfortunately, the currently approved therapies offer very limited efficacy, with median progression-free survival ranging from 2 to 4 months only. As Brian mentioned, the PAM, PI3K/AKT/mTOR pathway is one of the most important oncogenic pathways. It plays an important role in a number of critical cell functions, including cell metabolism, cell growth, proliferation, and survival. The PAM pathway also cross-regulates other oncogenic pathways and impacts immune-competent cells and tumor microenvironment. Multiple studies have demonstrated that PAM, hormonal, and cell cycle pathways play important roles in promoting tumor cell proliferation. Additionally, they are interdependent drivers for hormone-positive, HER2-negative advanced breast cancer, and this process is not dependent on PIK3CA mutation status. To maximize the efficacy, the evidence suggests that simultaneous blockade of all 3 pathways is very important. It is especially important to provide complete blockade of the PAM pathway. Doing so will prevent cross-activation of PAM pathway when both hormonal and cell cycle pathways are inhibited. It also can help to restore or enhance sensitivity to both endocrine and cell cycle targeting therapies. Because of this critical role in oncogenesis and ability to interact and cross-regulate other oncogenic pathways, we believe that PAM pathway represents one of the most important targets in oncology today. Let's please turn to slide number 10. Gedatolisib is a very potent inhibitor and very potently inhibit all four isoforms for class 1 PI3K and both mTORC1 and TORC2. This enables gedatolisib to provide complete and comprehensive blockade of the whole PAM pathway. This means that there is a limited potential for adaptive resistance when a single target inhibitor is used. For example, when someone targets just single isoform for PI3K, like alpha isoform, or just AKT or just mTOR. In several preclinical studies, gedatolisib inhibitor effect was 300 times more potent compared to those targeted a single PI3K isoform or AKT or mTOR. This potency was the same in tumor cell lines regardless of PIK3CA mutation status. Preliminary data with gedatolisib also found that patients with or without PIK3CA mutations had objective response rates well over 50%. Let's turn to slide 11. VIKTORIA-1 is a randomized, open-label global phase 3 study in patients with hormone-positive and HER2-negative advanced breast cancer. VIKTORIA-1 included patients with both PIK3CA mutation-positive and wild-type disease. Today, we are focusing on design and review of top-line results for Study 1, which included patients with PIK3CA wild-type disease. In this study, both men and women were eligible for enrollment. Patients must have had disease progression on or just right after treatment with CDK4/6 inhibitor in combination with nonsteroidal aromatase inhibitor. Up to 2 lines of hormonal therapy was allowed, and prior treatment with agents targeting PAM pathway was prohibited, as well as chemotherapy for patients advanced breast cancer. However, patients could have received chemotherapy as part of their treatment regimen in neoadjuvant or adjuvant setting. Patients also had to have measurable disease according to most recent RECIST version 1.1. In total, 392 patients were randomized equally 1 to 1 to 1 in 3 study arms. 2 of them were treatment study arms and 1 control study arm. Patients received standard doses of palbociclib in arm A and B. It's 125 milligram daily for 21 days with 7 days of treatment window. Fulvestrant in all 3 arms also was given as a standard dose of 500 milligram twice during cycle 1 and every 4 weeks thereafter. Gedatolisib was given in the standard dose of 180 milligram intravenously once weekly on days 1, 8, and 15. This 3 weeks on and 1 week off schedule provides patients with virtually 2 weeks break in treatment. For example, from day 15 to the day 1 of subsequent cycle, patient will have 2 weeks of break. In arm A, patients received triplet regimen of gedatolisib combined with fulvestrant and palbociclib. In arm B, patient received doublet regimen of gedatolisib combined with fulvestrant. And in a control arm C, patient received fulvestrant alone. It's important to notice that patients in arm C who had confirmed disease progressions were able to cross over and receive active treatment with either gedatolisib triplet or doublet regimen. The primary objective of this study was efficacy, assessed by comparing median progression-free survival between treatment arms. We had 2 co-primary endpoints. First one comparing efficacy in arm A versus arm C, and second arm B versus arm C. Let's turn now to slide 12. As Brian mentioned, both primary endpoints were met. Each shows statistically significant and clinically meaningful improvement in progression-free survival relative to fulvestrant alone. For patients in Arm A who received gedatolisib triplet regimen, median progression-free survival was 9.3 months, compared to 2 months for the patients in Arm C who received fulvestrant alone. This resulted in a hazard ratio of 0.24 and a very narrow confidence interval with a p-value less than 0.0001. This translates to 4.2 times higher chance of surviving without disease progression. These results are very clinically meaningful, with 7.3 months incremental improvement in median progression-free survival for triplet regimen. Let's now proceed to slide 13 and review results of the second co-primary endpoint. For patients in Arm B who received gedatolisib doublet regimen, median progression-free survival was 7.4 months, compared to 2 months for patients in Arm C who received fulvestrant alone. This resulted in a hazard ratio of 0.33 and a narrow confidence interval with a p-value less than 0.0001. This translates to a 3 times higher chance of surviving without disease progression for patients who received doublet regimen. These results are also very clinically meaningful, with a 5.4 incremental improvement in median progression-free survival for those who received gedatolisib in combination with fulvestrant. Let's turn to slide 14. Obviously, we're very excited. These efficacy results were so favorable. But we're also as pleased to report that both gedatolisib triplet and doublet were well tolerated, and the side effects were easily managed with available standards of care. The discontinuation of therapy due to treatment-related adverse events was lower in both gedatolisib triplet and gedatolisib doublet regimens, compared to those results observed in Arm B from our phase 1b study in advanced breast cancer that we presented in prior years. Additionally, the safety profile for both regimens was better than observed in the overall phase 1b study we presented earlier, including lower rates for stomatitis and hyperglycemia. Finally, the overall survival analysis shows positive trends and was favorable for both triplet and doublet regimens. Although the data is immature at this time, we are very encouraged about this because a very significant portion of patients in Arm C crossed over to receive active therapy, as I mentioned earlier. So patients in Arm C, upon disease progression, were able to receive either triplet or doublet gedatolisib regimens. The full results of VIKTORIA-1 PIK3CA wild-type cohort will be presented at the upcoming scientific symposiums later this year, and we are anticipating NDA submission in the fourth quarter of this year. We thank all patients, investigators, and site staff for participating in VIKTORIA-1 study. And I'm turning back to you, Brian.
Thank you, Igor. Let's turn now to slide 15. It's my pleasure now to introduce Dr. Rachel Lehman, Professor of Medicine at MD Anderson. Dr. Lehman provides a unique perspective. She was one of the top enrollers in both our phase 1b breast cancer study and the VIKTORIA-1 study. So she has a deep level of experience treating patients with gedatolisib. Let's turn now to slide 16. And Rachel, the floor is yours.
Thank you so much, Brian. Today, I'll be presenting the current treatment landscape for patients with hormone receptor positive, HER2 negative advanced breast cancer following prior treatment with endocrine therapy and a CDK4/6 inhibitor. And I'll be focusing on cancers without PIK3CA mutations. Unfortunately, after patients are treated with a first-line endocrine therapy combined with a CDK4/6 inhibitor, subsequent endocrine-based treatment regimens have not performed well. In an effort to improve outcomes for this patient population, multiple clinical trials of novel therapies have been performed. I will start by reviewing the data for novel endocrine therapy agents. This slide shows the oral SERDs, which have been in development for years. Although the expectations were high, phase 3 studies evaluating oral SERDs against standard of care endocrine therapy showed no significant benefit for cancers that do not harbor ESR1 mutations. The phase 3 studies with positive readouts are shown here. This data only includes the ESR1 mutation population, regardless of PIK3CA mutation status, as this is the population that benefited from therapy. The hazard ratios were similar for all three drugs, ranging from 0.55 to 0.66, with lower hazard ratios being more favorable. Elacestrant is the only oral SERD currently FDA approved. The EMERALD trial improved the median progression-free survival by only 1.9 months. In the VERITAC-2 study, vepdegestrant improved median progression-free survival by 2.9 months. And finally, the phase 3 EMBER-3 study enrolled patients with or without prior CDK4/6 inhibitor therapy. 30% of the study population did not receive a CDK4/6 inhibitor. Despite this, imlunestrant only improved progression-free survival by 1.7 months compared to standard endocrine therapy. Let's go to slide 17, please. Another approach to improve outcomes following first-line therapy was to continue a CDK4/6 inhibitor combined with a different endocrine therapy. The phase 3 post-MONARCH clinical trial randomized patients to receive fulvestrant with or without the CDK4/6 inhibitor abemaciclib. All patients received a prior CDK4/6 inhibitor combined with an aromatase inhibitor in the metastatic setting and had any PIK3CA and ESR1 status. The results were positive with a hazard ratio of 0.73. However, there was less than a 1-month improvement in progression-free survival with abemaciclib. The EMBER-3 study evaluated the oral SERD imlunestrant with and without abemaciclib. The study population in this trial was more diverse, with only 65% of patients receiving a prior CDK4/6 inhibitor, and the study enrolled patients in the first, second, and third-line treatment settings. EMBER-3 also yielded positive results with a hazard ratio of 0.57 and an absolute progression-free survival improvement of 3.9 months. Let's go to slide 8, please. Slide 18, please. Here we will move to the PAM pathway targeting therapy. To date, no prospective clinical trial in the post-CDK4/6 inhibitor setting has demonstrated benefit in cancers without a PIK3CA mutation. The most recently approved PAM pathway drug is capivasertib, an AKT inhibitor. However, no benefit was observed with the addition of capivasertib to fulvestrant in patients with PIK3CA wild-type breast cancer patients who had received a prior CDK4/6 inhibitor. Alpelisib is a PI3 kinase inhibitor, which is approved for the treatment of hormone receptor positive breast cancer with a PIK3CA mutation. The study leading to the approval of alpelisib was performed prior to wide availability of CDK4/6 inhibitors. and so most patients on this study did not receive a CDK inhibitor. Even so, patients without PIK3CA mutations did not benefit from the therapy and thus are excluded from the FDA approval of this drug. Everolimus is an mTOR inhibitor. Results from phase 3 studies were published more than a decade ago, prior to the availability of CDK4/6 inhibitors, among other therapies. Slide 19, please. Now let's look at how gedatolisib-containing regimens stack up against the current treatment landscape. First, I will show the median progression-free survival of the gedatolisib regimens, along with the approved regimens for which we have phase 3 data in the second-line setting. Again, for patients with hormone receptor positive, HER2 negative, and PIK3CA wild-type breast cancer, following CDK4/6 inhibitor therapy. Fulvestrant provides a progression-free survival of only 2 months, while elacestrant has a 3.9 progression-free survival when limited to the ESR1 mutant population. The 2 gedatolisib-containing regimens studied far outperformed the standard of care therapies with a median progression-free survival of 7.4 months when combined with fulvestrant and 9.3 months when combined with both fulvestrant and palbociclib. Slide 20, please. Now let's include regimens with available data that are not FDA approved at this time. This slide shows the absolute improvement in median progression-free survival with each regimen when compared to standard endocrine therapy. Gedatolisib clearly provides the highest incremental progression-free survival with a 5.4-month improvement with doublet therapy and a very impressive 7.3-month improvement with triplet therapy. Incremental improvement in the other regimens range from 0.7 to 3.9 months. Slide 21, please. This slide shows the same data shown by hazard ratio. The lower the hazard ratio, the better the treatment efficacy compared to control, which is standard endocrine therapy in this case. The hazard ratio for the triplet gedatolisib regimen is 0.24, and the doublet regimen has a hazard ratio of 0.33. These extremely favorable hazard ratios are rarely seen in oncology clinical trials and are unprecedented in this treatment setting. Up to this point, hazard ratios for this population in the second-line setting have ranged from 0.55 to 0.73. Slide 22, please. This data supports the potential for gedatolisib to become the standard of care drug for second-line hormone receptor positive, HER2 negative, and PIK3CA wild-type advanced breast cancer. Gedatolisib is highly efficacious. It is associated with the highest incremental improvement in median progression-free survival over endocrine therapy that has ever been reported in this patient population. The hazard ratio for triplet therapy of 0.24 and doublet therapy of 0.33 are the most favorable reported for any study in hormone receptor positive, HER2 negative advanced breast cancer. Additionally, gedatolisib is associated with preserved quality of life. Patients tolerated both gedatolisib-containing regimens well, with very low discontinuation rates. In addition, the safety profile for triplet and doublet therapy was more favorable than that previously reported in the phase 1b advanced breast cancer study. Of note, intravenous administration was not a barrier to use. Thank you very much. We can now go to slide 23, please.
Thank you, Rachel, for your very helpful summary of the treatment landscape. Let's turn now to slide 23. It's my pleasure to introduce Eldon Mayer, our Chief Commercial Officer. Eldon joined us last year, and he's done a great job of assembling a talented team and laying the groundwork for a potential commercial launch sometime next year. Eldon, please go ahead.
Thank you, Brian. Let's now turn to slide 24. I want to share a quick overview of the market landscape we see for gedatolisib, what we're doing to get ready, and how we're gearing up for a potential launch should we get FDA approval. So let's start with why we think the market looks promising for gedatolisib. First off, this is a sizable patient group. We estimate there are 34,000 patients moving to second-line treatment after CDK4/6 inhibitors, and roughly 60% of them are PIK3CA wild type. That's a very large opportunity. There's also a real need here. As we heard earlier on this call, current second-line treatments for HR-positive, HER2-negative advanced breast cancer are limited in terms of added progression-free survival benefit. Our market research shows that oncologists are hungry for options that are more effective and have a safety profile they can manage. With such a large underserved market, we see a chance to build a strong presence for Celcuity, and with gedatolisib's unique mechanism of action and corresponding clinical benefit, it is well positioned to address critical needs in this second-line space. And a couple quick points on market dynamics. IV-administered drugs, such as gedatolisib, fall under the medical benefit category, which means a typically smoother reimbursement process compared to oral drugs under pharmacy benefits, where payers tend to manage claims more heavily. Plus, the breast cancer community is active, engaged, and well supported by advocacy groups, which can help create awareness for new treatments. Based on our projections, this could represent a $5 billion served market revenue potential. Let's turn now to slide 25. All right, let's talk about how we're preparing for a potential launch in this market. Our experienced teams are hard at work getting ready, and we're on track for a strong market entry if we receive FDA approval. We've been gathering valuable insights from key opinion leaders, and now, with our clinical data in hand, we'll be investing more in those conversations to dig deeper into what these results mean for physicians and patients. Market access is a major focus for us to ensure patients have access to gedatolisib with a quick and seamless process when a physician orders it, and we're actively engaging with health systems, payers, pathway decision makers, group purchasing organizations, or GPOs. We're also working hard to educate and build excitement around the importance of comprehensively blocking the PI3K, AKT, and mTOR, or PAM pathway. That includes having a strong presence at key oncology conferences and continuing to educate and raise awareness about these issues within the breast cancer and oncology community. And we're tackling the major long lead time initiatives, such as setting up distribution, reimbursement, and building patient support systems, to name a few. So we're ready to execute these at launch. Let's turn now to slide 26. And finally, let's touch on our launch planning and infrastructure. With the teams we built, we're refining our strategic and tactical plans, especially now that we have phase 3 clinical data to work with. The most critical piece is having a seasoned, skilled team to execute, and we're ready to ramp up hiring quickly to make that happen. We're also staying close to oncologists through ongoing market research and working with key opinion leaders to ensure we're aligned with what oncologists and patients need, so we can position gedatolisib as effectively as possible. And we're making great progress on the operational side across our company, building out critical infrastructure such as supply chain, IT systems, data and analytics, field support, and much more to ensure operational excellence. All in all, we've made major strides in getting ready to become a commercial stage company, and we're pushing forward confidently to be fully prepared for a potential launch. Brian, back to you.
Thank you, Eldon. So let's now turn to slide 28, please. We have 3 important milestones coming up the back half of this year. First, we'll present the full data at a major medical conference later this year. Second, we expect to submit a new drug application, or NDA, for VIKTORIA-1 PIK3CA wild-type cohort indication in the fourth quarter of 2025. And finally, we hope to report top-line data for the PIK3CA mutation cohort of the VIKTORIA-1 study by the end of 2025. Let's turn now to slide 29. So this ends the presentation portion of our day. We'll turn now to the question and answer session. Thanks to all of you who did send in questions. And I should just mention that Rachel Lehman will have to attend to patients in just a few minutes, and we'll have to leave during Q&A. Hopefully, she'll be available for your questions. So let's open it up to questions, please.
Thank you. If you would like to ask a question, you will need to press star 1 and 1 on your telephone and wait for your name to be announced. And to withdraw your question, please press star 1 and 1 again. We will now start with our first question, and this is from Maury Raycroft from Jefferies. Please go ahead.
Hi, good morning. Much congrats on the data update, and thanks for taking my questions. Wondering if you can provide additional context to the 2-month PFS data observed in the control arm. Do you believe this patient population exhibits greater endocrine resistance compared to recent trials in the same setting? And what implications might this have for gedatolisib's effectiveness in a more sensitive population?
Thanks, Maury. So on one hand, we weren't surprised by the results for the fulvestrant arm. They've been, I think, 4 fairly recent clinical trials, prospective trials, randomized, that reported 1.9 to 2.1 months median PFS for fulvestrant. And there have been some outlier trials, but those results were really driven by differences in patient populations relative to a registrational study. So, this we think represents the current state and representative of what patients can experience in the setting. And so what that means, I think the translation is that these patients are not going to respond to much endocrine therapy, that essentially they're untreated disease mechanisms, which we think we demonstrated in this VIKTORIA-1 trial. As far as the sensitivity, we had data in our frontline, in our phase 1b study for endocrine sensitive patients. And in that study, it was preliminary data, but in a sample size of 41 patients, we reported median PFS of 48 months with gedatolisib, palbociclib, and letrozole. And so that's obviously very encouraging data. Our VIKTORIA-2 study, which we're currently enrolling, is focused on enrolling patients who are considered to be endocrine treatment resistant. And these are patients who progressed almost immediately after or during their treatment, endocrine treatment for early breast cancer. Big unmet need for these patients, that most of these patients are only getting therapies that offer them about 7 months median PFS. And so, we're encouraged by the results here. Obviously, we have to get the results for the VIKTORIA-2 study to see what the effect is. But certainly, the data we have today is encouraging for us and makes us optimistic about those future results.
Got it. That's helpful.
Oh, go ahead. Sorry.
Okay. Well, yeah, maybe one other question just on wondering if you could talk more about the favorable overall survival trend, what that means. Does that mean the trend's positive? And even though it's immature and with crossover, can you add any more context to how your OS data compares so far relative to competitors at the same stage?
Sure. Well, I think what was very encouraging to us was that patients with the triplet showed a favorable trend, even though in the control arm, a significant number of these patients, crossed over to receive gedatolisib therapy. And while favorable OS, positive OS is not required or hasn't been a benchmark that the FDA requires, they certainly expect to see no detriment to the overall survival trend for patients. And it was a recent study that reported encouraging progression-free survival data, but showed a fairly significant negative trend. And so, I guess everybody is more sensitive to the overall survival data as a result. And so in that context, we're particularly encouraged by what we've seen with the overall survival data.
Got it. Okay. Thanks for taking my questions.
You're welcome.
Thank you. We'll now take the next question. This is from Tara Bancroft, TD Cowen. Please go ahead.
Hi, good morning, and congrats on these fantastic data. So I was hoping you could talk more about the balance of the arms across mutational status and what consistency you might be seeing in those, especially in ESR1 mutant patients, and if you could compare those relative to EMBER-3 data specifically. Thanks so much.
Thanks. All that data, that type of data would be presented at later medical conferences. But what we can say is that the demographic data, baseline characteristics data that we've reviewed suggests that these arms were all well balanced. We had stratification variables, factors that ensured for critical factors that these populations or the arms were consistent and representative. So we don't expect to see any anomalies or anomalous accrual in according to mutational status or differences between arms.
Okay, great. Thanks. And then I guess for that full data, are you thinking more on the ESMO timeline or San Antonio timeline or something else?
Just stay tuned.
Okay, great. Thanks.
Thank you. We'll now take the next question. This is from Andrew Behrens from Leerink Partners. Please go ahead.
Hi, thanks, and congrats on the transformational results. Just wanted to ask a couple of questions on the future plans for the program. Can you just run through the rationale for the frontline trial, specifically the decision to target endocrine-resistant population with fulvestrant, just trying to put the results of the prior phase 1b in context with that program. And then it really seems to me a large opportunity could be to combine gedatolisib with the oral SERDs and to help those drugs beat fulvestrant in a broader population, which as the doctor ran through, none of those agents have been able to do it. So are you considering running those trials? It really seems like that could unlock a lot of fundamental and strategic value. Thanks.
Sure. Okay, thanks, Andy. So as far as the rationale for the first-line trial, we initiated work to get this trial going last year around middle of the year. And as we reviewed the unmet needs, we concluded that this population in particular was particularly underserved, and that with median progression-free survival outcomes in the first-line setting for most patients only being 7 months. And so from a practical standpoint, that means you can enroll a study and get data from that study in a fairly reasonable timeline. And so A, we're addressing an unmet need, and B, it just is a practical population to evaluate for a company at our stage of development. The endocrine-sensitive population gets a median PFS of roughly 25 months. And so it would be a much, much longer study. And one over time, we think would be appropriate to potentially go after because, again, the preliminary data we showed in the endocrine-sensitive population was very, very encouraging. And so VIKTORIA-2 then in some ways mirrors, you could say, the population we enrolled here, endocrine-resistant. And so, again, we're encouraged by these results, and we'll have to wait and see how that translates into VIKTORIA-2, but certainly gives us reason to be optimistic. And then as far as future development opportunities, certainly we think gedatolisib has shown that it can extend progression-free survival period when combined with hormonal therapy. And to the extent that there are better ways or different ways of offering patients an oral SERD, we want to certainly explore that. It'd be premature for us to really comment more specifically, but as we look longer term, we want to make sure that we position gedatolisib to be combined with as many appropriate drugs as possible, so that we give patients the best opportunity to get the best efficacy and safety profile possible.
Okay. Thanks, and congrats again.
Thank you. You're welcome.
Thank you. And we move to our last question today. This is from Oliver McCammon from LifeSci Capital. Please go ahead.
Congrats on the data, and thanks for taking my question. Just considering some of the baseline characteristics in VIKTORIA-1, like prior CDK4/6 inhibitor use, measurable disease, et cetera, can you remind us how heavily pretreated this patient population is versus some of the reference studies we've seen in the second or third line setting? I'm thinking specifically of things like EMBER-3 and CAPItello-291. Thanks again for the question.
Sure. Thank you, Oliver. Because of the enrollment criteria, we think, and we will disclose this data when we present the full data. But we clearly enrolled mostly a second-line population and some third-line patients. We did not allow patients who have had prior chemo in the advanced setting. Some of the other studies, for the most part, mirror this. To be frank, all these studies largely mirror this population. The EMBER-3 study had, as Rachel alluded to, a very diverse population. In some ways, you can think of them as having four subgroups of patients: first line, and then second line with or without prior chemo, or rather prior CDK. So it's really hard to kind of assess directly. But again, the hazard ratio comparison provides a way to kind of evaluate the relative contribution of the study drugs on top of hormone therapy. And so what's particularly interesting about this space is that for the most part, every study that's been run over the past, pick a number, 20 years, has essentially compared itself to endocrine monotherapy. And so you have the ability to kind of draw a bead on what the reasonable expectation is for these patients with endocrine monotherapy. And then what you'll see are some variations depending on whether patients are enrolled without measurable disease, which will happen in non-registrational studies, or patients may have received a more prior chemo. But for the most part, I think these populations really line up, except in cases where they're combined with first-line patients or patients who've not had CDK, and that really is a very small proportion of patients today. So really the most representative trials and the ones most relevant are ones that really isolated patients with prior CDK.
Super helpful, and thank you again. Congrats on the data.
Oh, you're welcome. Well, that wraps up our day. We appreciate you guys attending, and we look forward to providing these results in greater detail at an upcoming conference and to continuing our work to get ready for an NDA submission and to be able to get back with everybody with results from our mutant cohort. So look forward to chatting with some of you over the upcoming months. Thanks again.
Thank you. This concludes today's conference call. Thank you for participating, and you may now disconnect.